Hello everyone and welcome back to Biology with Dr. Soniyaa Shrivastava. In the previous session, we have done MCQs related to light reaction, photolysis of water, electron transport and non-cyclic photophosphorylation. But what happens when the plant needs extra ATP and how is ATP actually formed inside the chloroplast? So in today's class, we'll be covering headings 11.5 and 11.6 part 2 of chapter photosynthesis, in which we'll answer these questions by studying cyclic photophosphorylation and chemiosmosis, the two important concepts that are frequently tested in NEET. So let's begin. Let's start with MCQ number 1. Look at the chloroplast membrane diagram, at which site marked X is the oxygen evolving complex OEC located. Student, OEC is a metalloenzyme, consists of manganese and calcium ions along with oxygen atoms. Oxygen evolving complex is an water splitting machinery present in Photosystem II. Student, its location is very important because when water splits, that is, when photolysis happens, protons are released and the plants want these protons to accumulate inside the thylakoid membrane to create proton gradient for ATP synthesis. So before coming to the options, let's first see our diagram. Student, this is a diagram of non-cyclic photophosphorylation in which both the photosystem 2 and the photosystem 1 is present. In the photosystem 2 only, this X marks represent the oxygen evolving complex, which is the water splitting machinery in which the water split into protons, electrons and oxygen. Students, see here, stroma, which is the fluid film matrix, surrounds thylakoid membrane that is present in photosystem 2. This is the outer layer of thylakoid membrane and this is the inner layer of thylakoid membrane, which is known as lumen. Here present the OEC. See here, OEC is present inside the thylakoid lumen where water splitting occurs to give oxygen, protons and electron. These protons are released inside lumen. Now see our options. Option number 1, outer surface of thylakoid membrane. We know that the outer surface of thylakoid membrane faces the stroma. If water splitting occurs here, the released protons would enter into the stroma instead of lumen. So this option is wrong. Stroma matrix. The OEC is associated with photosystem 2, which is embedded in the thylakoid membrane, and it is not present in the stroma matrix. So this option is also wrong. Intermembrane space. The space lies between the outer and the inner chloroplast membrane and has no role in the water splitting. So this option is also wrong. Now the inner surface of thylakoid membrane. Yes, this is the correct option. We have seen the oxygen evolving complexes attached to the lumen facing, that is inner side of thylakoid membrane. This ensures that the released protons accumulate inside the lumen, which helps in ATP synthesis. Therefore, the correct option is option number 2. Let's see MCQ number 2. Cyclic photophosphorylation results in the formation of. Student, in the previous video, we have seen that photophosphorylation is of two types. First one is non-cyclic and second one is cyclic. We have already seen the non-cyclic process. Now the cyclic photophosphorylation. In the cyclic photophosphorylation, electrons leave the photosystem 1 and returns back to the photosystem 1 again. Since the electrons come back to where they started, the system works like a circular track. Therefore, this process is known as cyclic photophosphorylation. Now the big question is, what products can be formed in this cycle? But before that, we'll see this process in detail. Student, this is a diagram of cyclic photophosphorylation. This is the reaction center of Photosystem I. The reaction center chlorophyll absorbs the light at the wavelength of 700 nanometer. Therefore, it is also known as P700. Let's see the process. When light falls on the photosystem I, that is P700, electron gains the energy and excited to primary electron acceptor. See here, when the light falls on the reaction center, electron gains energy and excited to primary electron acceptor. Then from primary electron acceptor, electron goes to ETC or ETS, that is Electron Transport System, which consists of different carriers. First one is ferrodoxin, which is the iron containing carrier. From there, electron goes to second carrier, that is cytochrome. While passes through cytochrome, electron loses some energy, and this energy is used by cytochrome to pump the protons to form the proton gradient to synthesize ATP. Then from cytochrome, electron goes to third carrier, that is plastocyanin. And from plastocyanin, electron comes back to photosystem I again. And in this process, there is no water splitting machinery, no NADP reductase to reduce the NADP plus to NADPH. Here only present ATP synthase to form ATP. Let's see our option number 1. ATP and NADPH. We know that ATP is formed, but what about NADPH? NADPH is not produced in the cyclic photophosphorylation because the electrons never reach the NADP+. They return to PS1 only. Therefore, this option is wrong. Now option number 2, ATP, NADPH and O2. We already know that these are the products of non-cyclic photophosphorylation, because in case of cyclic photophosphorylation, there is no water splitting happens, so no oxygen is released, so no NADPH is formed. This option is wrong. ATP only. Yes, this is our answer. As the electron cycles through electron transport chain, energy is released and used for ATP synthesis. ATP is the only product in case of cyclic photophosphorylation. So this is our correct option. NADPH we already know that requires electrons to reduce NADP+, but in cyclic flow electron returns to PS1 only and never reduce NADP+. Therefore, this option is also wrong. Moving on to MCQ number 3. The stroma lamellae membranes lack. Student, before coming to the options, let's first revise our concept. Student, remember the structure of chloroplast. In the structure of chloroplast is present the stacks of thylakoids. They are known as grana. And these stacks of thylakoids are connected with each other by a thin membrane known as stroma lamellae. Stroma lamellae are the bridges connecting the stacks of thylakoid called grana, and they are present inside the chloroplast. They both are membranous structure. Then also stroma lamellae are not identical with grana. There are something missing from stroma lamellae. As we know that grana is the main light harvesting complex, PS2 is placed here. Presence of PS2 means presence of NADP reductase enzyme. That means reduction of NADP plus to NADPH apart from formation of ATP and O2 as byproduct. So they are mainly performing non-cyclic photophosphorylation, but stroma lamellae lacks PS2. That means it also lacks NADP reductase enzyme and it has PS1. That means only the formation of ATP through ATP synthase. That means cyclic photophosphorylation. Coming to our options, option number 1, PS1 and NADP reductase. But PS1 is present in stroma lamellae, therefore this is wrong option. Option number 2, PS2 and NADP reductase. Yes, this is our correct option. PS2 and NADP reductase are absent in stroma lamellae. Therefore, this is our correct option and they are present only in grana. PS1 and PS2, as PS1 is present in stroma lamellae, so this is also the wrong option. ATP synthase is also present in stroma lamellae, so this is also the wrong option. Student, there is a quick memory table by which you can remember easily everything like component, grana and stroma lamellae. Components are PS2, PS1, NADP reductase and ATP synthase. PS2 in case of grana is present, in case of stroma lamellae is absent. PS1 is present in both. NADP reductase is present in grana but absent in stroma lamellae and ATP synthase is present in both. So in this way you can remember easily what is present in grana and what is present in stroma lamellae. Moving on to MCQ number 4. When only light of wavelength beyond 680 nm is available, which process will occur? Beyond 680 nm means more than 680 nm. Student, before coming to the options, remember that Photosystem II has reaction centered chlorophyll that absorbs light at 680 nm, while Photosystem I has reaction centered that absorbs light at 700 nm. If the available light has wavelength beyond 680, that means more than 680, so PS2 cannot function efficiently, that means non-cyclic photophosphorylation stops. So the question is asking what process can continue when only PS1 remains active. So let's see our options. Non-cyclic photophosphorylation. Non-cyclic photophosphorylation requires both PS2 and PS1 working together. If PS2 is inactive, non-cyclic pathway cannot operate. This option is wrong. Cyclic photophosphorylation. Yes, this is our correct option. Cyclic photophosphorylation involves only PS1. Since PS1 can still absorb long wavelength above 680 nm, only the cyclic photophosphorylation occurs. So this is our correct option. Photolysis of water. Water splitting is associated with PS2 and PS2 occurs in 680. So beyond 680 it becomes inactive. So this option is also wrong. Evolution of oxygen is related with photolysis. That means PS2 and 680. So this option is also wrong. Moving on to MCQ number 5. In the flow diagram of cyclic photophosphorylation, which component is missing compared to non-cyclic? So before coming to the options, let's first see its flow diagram. This is the flow diagram of cyclic and this is non-cyclic one. Student, non-cyclic photophosphorylation is the complete setup as it involves both the photosystem, Photosystem II and Photosystem I, and the production of ATP in cytochrome through enzyme ATP synthase and production of NADPH by the reduction of NADP plus through enzyme NADP reductase, and third, oxygen as byproduct by the splitting of water. While cyclic photophosphorylation is a simplified pathway that involves only PS1 and cytochrome B6F, ATP production through enzyme ATP synthase. So what is missing in the cyclic process as compared to non-cyclic one? So coming to the options. Option number 1, PS1 is present in cyclic process. So this option is wrong. ATP synthase is also present in the cyclic process. So this option is also wrong. Option number 3, PS2 and NADP reductase. Yes, this is our correct answer because these two are absent in cyclic process and present in non-cyclic one. So this is our correct option. Cytochrome B6F, this is present in both the cases, cyclic and non-cyclic. Therefore, this is also the wrong option. Moving on to MCQ number 6. According to the chemiosmotic hypothesis, ATP synthesis is linked to the, first of all student, what is chemiosmotic? Chemiosmotic is the movement of ions, mainly H plus ions or protons, across the selectively permeable membrane. Permeable membranes means the membrane which allow liquid or gases to pass through their pores. So chemiosmotic means the movement of ions, mainly hydrogen ions or protons, across selectively permeable membrane down their electrochemical gradient. Now, chemiosmotic hypothesis explains how cells generate energy in the form of ATP. Student, we have seen the two processes, that is cyclic and non-cyclic photophosphorylation, but we didn't actually know how ATP is synthesized in these two processes. So, in this question, we'll also see how ATP is synthesized. So, before coming to the options, let's first see our concept. We have already know that chemiosmotic hypothesis explains how cells generate energy in the form of ATP. Now, during the light reaction, proton comes from water splitting, which releases protons to lumen. Second, cytochrome B6F complex pumps more proton into the lumen of thylakoid membrane. Third, NADP plus reduction removes protons, that is H plus, from the stroma. So during light reaction, these three are the process through which proton comes into the lumen. As a result, what happens? This is stroma and this is thylakoid membrane and this is inner side of thylakoid membrane, that is lumen. So because of all these three processes, stroma has low hydrogen ions because these three processes pumps the proton towards the lumen. So lumen has more H plus ions. Now this difference in hydrogen ion concentration across the thylakoid membrane develops proton gradient. Now, due to this imbalance, proton wants to go back to stroma. Again we'll see stroma, thylakoid membrane, and in thylakoid membrane embedded a ATP synthase enzyme. So here is low H plus and here is lumen more H plus. Due to imbalance, proton wants to come back to the stroma, so they pass through the ATP synthase. As the proton flow through ATP synthase uses energy from proton gradient to attach Pi, free inorganic phosphate, which is present in the stroma, to ADP. That is ADP plus Pi. This is adenosine diphosphate. One more inorganic phosphate is attached to ADP to form adenosine triphosphate, ATP. Now option number 1, development of electron gradient. Student, this is a trap. Electron moves through ATS, but ATP synthase not driven by electron gradient. It is driven by proton gradient. So this option is wrong. Now option number 3, reduction of glucose. Glucose is synthesized during the Calvin cycle, that is in dark reaction, not in the light reaction. It is not the direct driving force for ATP synthesis. So this option is also wrong. Oxidation of NADPH. NADPH is mainly used during carbon fixation in the stroma. Its oxidation is not the basis of ATP formation. So this option is also wrong. Now the development of an proton gradient. Yes, this is our correct answer. Why? We have already seen as electron moves through ETS, that is in cytochrome, protons accumulate inside the thylakoid lumen. When these protons flow back through ATP synthase from lumen to stroma, ATP is produced. Student, let's see MCQ number 7. In the chemiosmotic diagram, toward which side does the F1 head of ATP synthase protrude? Student, ATP synthase looks like a tiny turbine embedded in thylakoid membrane, as we have already seen in the previous MCQ. Protons accumulate inside the lumen of thylakoid membrane and flow through ATP synthase towards the stroma. So the ATP form is released into the stroma, and this ATP is used in the Calvin cycle, that is dark reaction, which occurs in stroma. Student, this gives us big clue about the orientation of ATP synthase, that where does the F1 head of ATP synthase protrude. Now before coming to the options, let's first see its diagram. Student, this is the diagram of non-cyclic photophosphorylation, and this is ATP synthase. See here, ATP synthase. ATP synthase has two major regions or parts, that is F0 and F1. Now let's see its flow diagram. Stroma, then F1 head, ATP synthase, thylakoid membrane, F0 ATP synthase. F0 regions embedded in thylakoid membrane. Secondly, it forms the channel through which protons pass from lumen towards stroma. Now F1 head is projected towards stroma and it contains the catalytic site and synthesize ATP from ADP plus Pi. Therefore, the F1 head of ATP synthase protrudes into the stroma. allowing ATP to be synthesized exactly where the Calvin cycle uses it. Now we'll see our options. Number 1 is thylakoid lumen. The lumen is where the protons accumulate before flowing through ATP synthase. However, the ATP producing F1 head is not located here. This is the location of F0 of ATP synthase. Therefore, this is the wrong option. Stroma. Yes, this is the correct answer, because the F1 head of ATP synthase projects into the stroma. Inner membrane space. This term is associated with space between chloroplast membranes, not with ATP synthase orientation in the thylakoid membrane. So therefore, this is also the wrong option. Cytoplasm. ATP synthase is involved in photosynthesis, is located inside the chloroplast, not facing the cytoplasm. Therefore, this option is wrong. Let's see MCQ number 8. During the light reaction, protons accumulated in the. Student, we have already seen in the previous MCQs that during light reaction, where the protons gets accumulated, we have seen that the protons are coming from splitting of water, pumping from cytochrome, and through reduction of NADP plus, and accumulated in the thylakoid membrane, but not on the outer surface, but in the lumen, that is inner surface of thylakoid membrane. Now see our options. Option number 1, stroma. No, protons are not accumulated here. In fact, the protons are removed from the stroma and pumped away during the light reaction. So this option is wrong. Intermembrane space of chloroplast. This is the trap. In chloroplast, proton accumulate in the thylakoid lumen, not in the intermembrane space. So this is the wrong option. Lumen of thylakoid. Yes, this is our correct answer. Protons are released into and pumped into the thylakoid lumen. Cytoplasm. Cytoplasm is not involved in the proton accumulation for photophosphorylation. So this is the wrong option. Moving on to MCQ number 9. The enzyme ATP synthase consists of two parts. The F0 part is. Student, in the previous question, we have already seen that ATP synthase has two parts, that is F0 and F1. Now before coming to the options, let's again revise our concept. We have already seen that stroma, that is fluid filled matrix. Here is F1 head of ATP synthase, and this is thylakoid membrane, and here is F0 channel of ATP synthase, that is embedded in lumen of thylakoid membrane, where protons are accumulated. F0 acts as channel for passing protons, while F1 head is responsible for ATP synthesis. In the previous question, we have seen that F1 head is projected towards the stroma. Then what about F0? F0 is embedded inside the thylakoid membrane and act as a proton channel. Now coming to our options. Option number 1, located in the stroma. No, the part facing the stroma is F1. So this is not our option. Embedded in the thylakoid membrane. Yes, option number 2 is correct. F0 part is embedded in the thylakoid membrane. Option number 3, a peripheral membrane protein. This describes F1, which is attached to the membrane surface and protrudes into the stroma. So this is also not our option. Responsible for sugar synthesis. Sugar synthesis occurs in the stroma during the Calvin cycle. So this is also the wrong option. Moving on to MCQ number 10. Which of the following does not contribute to the proton gradient. Student, we know there are three factors which create and strengthen the proton gradient across the thylakoid membrane. But the question is asked, which option does not help build this gradient? So let's see our options. Option number 1, splitting of water on the inner side of membrane. Yes, this contributes. How? When water is split at pH2, protons are released directly into the thylakoid membrane, increasing the hydrogen ion concentration there. So this is not our option. Option number 2, pumping of protons by cytochrome complex. Yes, this also contributes. As electrons move through cytochrome base 6F, protons are actively transported from stroma to lumen, strengthening the gradient. So this is not our option. Option number 3, reduction of NADP plus to NADPH on the stroma side. Yes, this also contributes. During NADPH formation, protons are consumed from the stroma. So fewer ions in the stroma makes the difference between lumen and stroma even larger. Therefore, this option is also wrong. Now option number 4, movement of electrons from pH2 to phosphatin. Phosphatin is the primary electron acceptor. Electrons are simply transferred from pH2 to primary electron acceptor, that is phosphatin. No protons are pumped or accumulated during this step. So it does not directly contribute to the proton gradient. So this is our correct option. Let's move on to MCQ number 11. This is assertion reason based question. What is assertion given here? That ATP is produced when protons move across the membrane through the F0 channel of ATP synthase. What is reason given here? This movement causes a conformational change in the F1 head of ATP synthase. Student, ATP synthase works like a tiny turbine. Protons flow through the F0 channel just like a water flows through the hydroelectric dam. But ATP is not made by the flow itself. The flow causes structural changes in the F1 head, and those changes drives the ATP synthesis. So before coming to the options, let's see our assertion and reason, and most important, connection between them. So what is assertion given here? That ATP is produced when protons move across the membrane through F0 channel. Is this statement is true? Yes, this statement is true. How? Let's see our flow diagram. This is stroma, and here present the F1 head of ATP synthase, and this is thylakoid membrane, and here present the F0 channel of ATP synthase. Protons are accumulated here. According to chemiosmotic hypothesis, protons move down their gradient through F0 channel of the ATP synthase, providing energy needed for ATP formation. Now what is the reason given here? That this movement causes conformational change in the F1 head. Is this statement is true? Yes, this statement is also true. The energy released by the proton flow causes changes in the shape of F1 head, which catalyzes the conversion of ADP to ATP. So both these statements are true. Now does the reason explain the assertion? Yes, reason explains the assertion. ATP is produced Because the proton movement through F0 causes the conformational change in the F1 head, and those changes are the actual mechanism of ATP synthesis. So coming to the options, both the statements are true and the reason is the correct explanation of A. So the correct option is option number 1. Now let's see MCQ number 12. The NADP reductase enzyme is located on student. NADPH is the reducing power used in the Kelvin cycle, that is the dark reaction that occurs in the stroma. So the question is asking where should the enzyme NADP reductase be located that makes the NADPH. So before coming to the option, let's see our flow diagram. Stroma, NADP plus reduced to NADPH, NADP reductase, thylakoid membrane, lumen. This flow diagram shows that NADP reductase enzyme is present on the stroma side. Now coming to the options. Option number 1, the lumen side of thylakoid membrane. Is it so? No. If the NADPH were produced inside the lumen, it would have to cross the membrane to reach the Kelvin cycle enzymes in the stroma. So this option is wrong. Option number 2, the stroma side of the thylakoid membrane. Yes, this is the correct answer, as we have seen that NADP reductase is located on the stroma side. Why? Because it reduces NADP plus to NADPH. Since NADPH is required in the stroma for the Kelvin cycle, the NADP reductase enzyme is strategically located on the stroma side of the thylakoid membrane. Option number 3, the outer chloroplast membrane. No, this option is wrong. NADPH production is the part of light reaction which occurs in the thylakoid membrane, not the outer chloroplast membrane. So this option is wrong. The mitochondrial cristae. This is wrong originally. Cristae belong to mitochondria, whereas NADP reductase functions in the chloroplast. Moving on to MCQ number 13. During high intensity photosynthesis, the pH in the thylakoid lumen. So before coming to the option, let's first see what is pH. pH is the potential of hydrogen or hydrogen ion concentration or protons. It is the measurement used to determine how acidic or basic the solution is, and pH scale typically ranges from 0 to 14, where 7 is neutral. Below 7, the solution is acidic because of the presence of high concentration of hydrogen ions. That means they are having the low pH. And above 7, the solution is basic because of the presence of higher concentration of hydroxyl ions. This shows the high pH. If we see the flow diagram of chloroplast, because of the light reaction 3 processes, hydrogen ions are pumped into the lumen of thylakoid membrane. So here more protons are accumulated compared to stroma. This shows that the lumen pH is acidic, means low pH, as compared to stroma. Now coming to our options. Option number 1, increases. This is the trap. An increase in pH means fewer hydrogen ions. But during active photosynthesis, hydrogen ions are more in the lumen. So this option is wrong. Option number 2, decreases. Yes, this is our correct answer. Why? Because as more protons accumulate in the lumen, it becomes more acidic, showing low pH. Remains neutral. No, this is wrong. The lumen does not stay neutral during active photosynthesis. It becomes acidic due to proton accumulation. So this is wrong. Becomes equal to stroma pH. If the lumen and stroma had the same pH, there would be no proton gradient. So no ATP synthesis. So this option is also wrong. Moving on to MCQ number 14. In chemiosmosis, the energy required to create proton gradient comes from students' protons do not move into the lumen by themselves. Pumping hydrogen ion against the concentration gradient requires energy. The question is, where does that energy come from? So let's see our concept first. We know when light hits the photosystem II, electron gets excited, and the excited electron moves through ETS, that is Electron Transport System, which consists of different carriers. As the electron moves downhill through carriers, energy is released, and that energy is used to pump the proton to thylakoid lumen, creating proton gradient to synthesize ATP. Now coming to the options. Option number 1, the breakdown of glucose. This happens during respiration, not during the light reaction of photosynthesis. So this is the wrong option. Option number 2, the hydrolysis of ATP. ATP hydrolysis releases energy, but here we are trying to make ATP, not spend it. So this is also the wrong option. Option number 3, the movement of electrons through the ETS. Yes, this is our correct answer. As electrons flow through the electron transport chain, energy is released, and this energy powers the pumping of hydrogen ions into the lumen, creating proton gradient to synthesize ATP. The absorption of CO2. CO2 is used later in the Calvin cycle for sugar synthesis. It does not provide energy for proton pumping. Therefore, this option is wrong. Moving on to MCQ number 15. Which of the following is not a requirement for chemiosmosis to occur? We know that chemiosmosis needs a few essential components to make ATP, and the question is asking which is not that component. Let's see our option. Option number 1, a membrane, thylakoid membrane. The membrane separates the lumen from stroma and prevents protons from simply diffusing away. Without a membrane, no gradient can be maintained. This is essential in chemiosmosis. Therefore, this is wrong option. Option number 2, a proton pump. The proton pump uses energy from electron transport to move protons into the lumen and build the gradient. Therefore, proton pump is also essential. Therefore, this option is also wrong. Option number 3, proton gradient. ATP synthase works only when protons flow down a concentration gradient. No gradient means no ATP production. So, this is also essential in chemiosmosis. Therefore, this is also the wrong option. Option number 4, NADP reductase enzyme. NADP reductase is important for making NADPH during the light reaction. But chemiosmosis itself does not require this enzyme. ATP synthesis can occur without NADP reductase also. This is our correct answer. With this, we complete the light reaction of photosynthesis and understand how ATP is synthesized through chemiosmosis. In the next part, we will study the Calvin cycle, where these molecules help convert carbon dioxide into sugars, and we'll also explore the C4 pathway, a special adaptation that makes photosynthesis more efficient in certain plants. Thank you, and I will see you in the next session.